This is a very low power view of a classical liver lobule. Characteristic landmarks of the classical liver lobule are the central vein and the portal area. Now, as you may know, the portal area contains a branch of the hepatic artery, a branch of the portal vein, and a branch of the bile duct. Now, we will obviously talk about liver histology later in the course, so this slide is right now here just to help you get your bearings as you look at the liver. This view shows a little bit higher power view of the portal area, and here you can see the structures labeled. Note the arrow for the hepatic artery is in the lumen of the artery, the arrow for the portal vein is in the lumen, and the arrow for the bile duct is in the lumen. On the hepatic artery, there's a single layer of endothelial cells that line the lumen, and there are several layers of smooth muscle, very typical of an artery. The portal vein also has a single layer of endothelial cells lining the lumen, very little smooth muscle. And note that the bile duct now has sort of a cuboidal epithelium lining its lumen. Again, we'll talk about the histology of this later in the course. At this point, it's just for you to get your orientation. Now, the material in the lumen of the hepatic artery is simply red blood cells and maybe some coagulated protein material. It's a preparation artifact. Here we'll back down in magnification to the classic lobule so you can see the position of the central vein again. In higher magnification, you can see the central vein, and you can see rows of hepatocytes that radiate out from the central vein, almost like spokes on a bicycle wheel. And the white spaces between these rows of hepatocytes are actually the liver sinusoidal spaces, and these spaces are continuous with the luminal space of the central vein. At higher magnification, you can see the sinusoidal spaces on either side of the plates or rows of hepatocytes. Every now and then you can see some endothelial cells that make up part of the wall of the sinusoidal space. We will, of course, talk about sinusoidal capillaries later in the course. For now, you should understand that by virtue of the sinusoids, hepatocytes are essentially bathed in plasma. Now, something not readily visible on this image, but highlighted nevertheless by the black arrows, would be bile canoniculi, and these would be spaces into which the hepatocytes are secreting bile. This is a diagram of a single hepatocyte. The yellow space would be equivalent to the sinusoidal space, and you can see the green areas would be equivalent to the bile canoniculi. Now, it's important in your understanding of the function of hepatocytes to realize that they secrete bile into a space that's separate from that space that bathes the hepatocytes in plasma, which on the diagram here again would be everything shown in yellow. On this diagram, you can see the large endothelial cells, and below those would be the luminal space of the sinusoids. Above the endothelial cells, you see something labeled space of Dissay. We will talk about this later in the course when we talk about the histology of the liver. What I'd like you to realize, the space of Dissay is essentially space that's equivalent to plasma space, albeit there are no formed blood cell elements in the space of Dissay. But I think you should be able to understand from the diagrams that you can clearly see that hepatocytes are totally bathed in plasma except at the areas where there are bile canoniculi. Now, you should look at the electron microscopic atlas of hepatocytes to get a better appreciation of the different cell surfaces on hepatocytes and to get some appreciation of how the hepatocytes begin to function at the cell biological level.